Hello class, Dr. Pavlovic. My name is Sam Shakespeare, and my topic briefly explores the possibility of growing complete transplantable organs from stem cells. Ever since Shinya Yamanaka in 2006 discovered that adult stem cells could be reprogrammed to their embryonic pluripotent state, research in many areas of stem cell therapy has exploded. Because, as we learned in this class, induced pluripotent stem cells can develop into any other cell types, tissues, or organ. iPS cells also have the capability to self-renew, reproducing themselves many times over, and because iPS cells can be made from a patient's own adult skin cells, that can be used to grow specialized cells and tissues that exactly match the patient and would not be rejected by the immune system, indicating great potential for new applications in medicine. One of these applications could be to regenerate complete transplantable organs from a patient's own stem cells. Today, there are over 120,000 patients awaiting organ transplants. These patients may have to wait weeks, months, or even years until an organ becomes available to them, and many of these candidates will die before an organ becomes available. An interesting fact is that each day an average of 79 people receive organ transplants. However, an average of 22 people die each day waiting for transplants because of the shortage of donated organs. If cultivation of complete transplantable organs becomes a reality, it will reduce, if not eliminate, the need for donated transplanted organs, which carry the risk of immune system rejection. Currently, regrowing a complete organ like a lung or liver or heart has not yet been achieved, but scientists have successfully grown a range of human biological structures and organoids, and many from induced pluripotent stem cells. So what are organoids, you ask? They are mini organs grown in vitro that mimic some of the structure and function of real organs. Some examples include a team from Japan succeeded in generating mini livers from iPS cells taken from human skin. When mixed with two other types of cells, the stem cells organize themselves in three-dimensional structures complete with blood vessels. Another example involves the Cincinnati Children's Hospital Medical Center, where iPS cells were used to grow human intestinal tissue in a lab. That tissue was then connected to a kidney in a mouse, providing it with the blood supply that allowed it to mature into a piece of human intestine. This could be a useful way of studying gastrointestinal diseases in the future. Progress in regenerative medicine is being made every day, and important research is being conducted now in Australia that has produced kidney organoids from iPS cells. The research team bathed the stem cells in chemicals to force them to differentiate into kidney cells. The stem cells began to differentiate into various kidney tissue types, and more importantly, they began to self-organize into a kidney-like structure. This ability for stem cells to self-organize provides an opportunity for us to address the complexity of tissue structures in regeneration. Here's an image of the self-organized kidney organoid formed in a dish from human-induced pluripotent stem cells. Now, this is not a complete kidney, but an organoid. There's still a long way to go until clinically useful transplantable kidneys can be engineered, but this is a step in the right direction. These organoids will prove invaluable in the ability to test drug safety on human kidney tissue instead of testing on animals that may not provide accurate results. To re-emphasize, organoids are not fully functioning organs, but they do represent an important step in the successful cultivation of complete transplantable organs. They may also satisfy an important medical need, and that is in drug safety testing. The cultivated organoids are instrumental in observing the effects of certain drugs on human tissues. We've also seen that organoids leverage the self-organizing properties of stem cells to create diverse, multicellular tissue structures. Organoids can be grown in petri dishes, but to cultivate a complete three-dimensional organ, we need a suitable support structure. This is called a scaffold. The scaffold is a three-dimensional support biostructure combined with a delivery system for growth factors that provide a foundation for the stem cells to grow in the shape of the desired tissue or organ. Here are some examples of scaffolds that have been used in medical applications. You can see here an image of a biomanufactured trachea, as well as a few images of scaffolds that would help in reconstructing the human ear. The scaffold needs to be carefully constructed as it holds the biomaterials in place and allows the living tissue to live and regenerate. With the introduction of 3D printing, Research into printed scaffolds and tissues has continued to develop. Meet Dr. Anthony Atala, director of the Wake Forest Institute for Regenerative Medicine in North Carolina. He and his team have been using iPS cells to grow muscles, blood vessels, skin, and even a complete urinary bladder, which after being laboratory grown was implanted into more than 24 children and young adults born with defective bladders. The bladders were made by growing the patient's iPS cells in a petri dish for several weeks, creating enough muscle cells for an entire bladder. The cells were then poured over a collagen scaffold with some other materials and nurtured in an incubator that mimics body conditions. Once the bladder is implanted into the patient, the scaffold gradually dissolves. Dr. Atoll is using custom-designed 3D printers equipped with special bioinks that allow for simultaneously printing the scaffolds along with the stem cells that will later make up the tissue. Sometimes 3D printing cannot do the entire job. Currently, the heart is too complex of an organ to be completely printed by a machine, so another method must be used, and this one uses a natural heart scaffold, a process called decellularization. Decellularization is when a donor organ, unsuitable for transplantation, is submersed in detergent solutions that strip away any cells that could trigger a recipient's immune response. This process leaves behind the extracellular matrix, or ECM. The ECM is a non-cellular component present within all tissues and organs and provide essential physical scaffolding for the cells and tissues. Considering that there are over 4,000 Americans waiting for a heart transplant and heart disease is being the #1 cause of death for men and women, the question must be asked, is it possible to grow a human heart? Well, teams from Harvard Medical School and Massachusetts General Hospital are asking that question right now. They took 76 hearts deemed unsuitable for transplantation and decellularized all of them, leaving only their protein scaffolds behind. These scaffolds contain the intricate structures and vessels that would provide the new foundation for new heart cells to grow into. The researchers then reprogrammed human skin cells into becoming iPS cells and forced them into becoming 2 types of heart cells. Within just 2 weeks, in a bioreactor, simulating the heart's natural environment, the networks of lab-grown heart cells already resembled immature but intricately structured hearts. And what's amazing to me is that when the team administered a burst of electricity, all 76 hearts started beating. Now, these immature hearts contain about 500 million heart cells each. That's a far cry from a fully mature heart, which has closer to 10 billion heart cells. So clearly, we are still a long ways away from growing complete organs ready for transplant, but progress is being made every day. I wonder how long it will be before we grow full limbs. Thanks for listening. Have a great day.